Welcome back, friends. This is Dr. Mlelwa Lecturers, the facilitator in this session. I am Dr. Mlelwa, and in this session we are launching the session in the advanced and the O-level biology, chemistry, mathematics and physics. So in today's session we are discussing about the Form 3 chemistry, and we are going to discuss about the determination of molality. Before discussing about the calculations of determination of molality, we touch a little about the choice of indicators during acid-base titration. Now, if you remember, in the previous session we discussed about the preparation of standard solution of acids. Tuliona ni kwa jinsi gani kama tumepewa stock sulfuric acid, hydrochloric or nitric acid, we can prepare the standard solution. So we need to do different calculations, and after calculating those calculations we need to know how we measure the volume, and after measuring volume we need to know how we can mix that volume with water using certain principles in order to avoid the corrosive reactions which can injure our skin in the laboratory, and thus avoiding the unnecessary accidents in the laboratory. Now, in today's session, as I told you, that we are going to discuss about the concept of calculation of molar solution or molarity. So what we are saying is that determination of molarity, or sometimes we call it as standardization of sulfuric acid solution by titrating against the 0.1 sodium carbonate solution. So one thing you need to know here that when we have two solutions here. We have the acid, let's call it as IA, and we have the base, let's call it as B. So for our case, our acid is sulfuric acid, H2SO4, and for our case our base is sodium carbonate, that is sodium 2 CO3. This is for our case. Now we have the acid here, and then we have the base here. In volumetric analysis, the concentration of one among these must be known. Hii tuseme tunafanya standardization. Standardization means we are using the molarity of a known solution, let's say base, to calculate the molarity of a known solution, let's say acid. So the molarity of known solution, the molarity of known solution, as we said previously, that if the solution had the known molarity, it is termed as the standard solution. Or sometimes in standardization we can use the molarity of known solution, let's say acid, to calculate the molarity of a known solution, let's say base. Now if this is the solution with known molarity, now this will be called as the standard solution. So a standard solution can either be an acid or base, depending on different conditions. But all in all, a standard solution is the solution whose concentration is exactly known. That's all about the concept of standardization. It's just a part of introduction of standardization. When you are saying standardization, what you mean? Standardization is we are using the known solution, the concentration of known solution, or we are using the solution with known concentration to calculate the concentration of a solution with the unknown concentration. Kwa kuna solution moja ambayo concentration yake is unknown. We are using the known concentration to calculate the unknown one. So what we are saying ni kwamba kwenye hiyo concept tunasema kwamba estimation of concentration of solution for an acid by reacting it, we are estimating the we are estimating the concentration of a solution of an acid by reacting it with the base, by reacting it. So we are saying it is by reacting the acid with a standard alkali solution is known as titration. So we are using the acid ambayo tujui concentration yake, then we are titrating it with the standard alkali solution. So the end point of an acid-base reaction is commonly determined by using a substance known as indicator. Very important concept for you to understand when you are discussing about the concept of acid-base titration. One among the very important concept for you to understand is about the concept of indicator. So what is an indicator and how it functions, we study it in detail later. So firstly, by definition, titration is the process of adding an acid to a base until an indicator shows that the reaction is complete. The reaction is complete. So this is the definition of titration, or the definition of acid-base titration. Now what is indicator? Indicator the chemical substance which change color when the end point of a titration reached, or with the chemical substance which change color when the pH of the medium changes. Kwa sababu kinachosababisha the indicator to change color is the change of the pH. What is the pH? Hiyo tunasoma kwenye acid base and salt. Kama hujasoma, I will discuss it later. It is also the form 3 chemistry topic. Kwa hiyo tunachokisema ni kwamba if we have the acid and then you have the base, titrating them will reach a certain concentration where the acid and the base amount in the solution will be equal and the solution will be regarded as neutral. So the change in the pH will lead to the change in the indicator color, and at the end point the pH is neutral. That means all the acid which is present in the solution has been reacted with the base and the pH has become to neutral. If the pH becomes to neutral, then the color becomes, the color of the indicator changes. So one among the very important point also to consider when you are discussing about these things is about the choice of indicator, because when we are titrating a solution of different concentration, we are choosing different kinds of indicator. For example, when we are titrating sodium hydroxide, let's say it is sodium hydroxide, titration of sodium hydroxide, sodium hydroxide. So it is titration of sodium hydroxide plus, let's say it's HCl, sulfuric acid, dilute sulfuric acid. So what indicator will you use? You need to know the choice of indicator. In order to know the choice of indicator, you need to know about the nature of an acid and a base which is used in that titration. So for example, if you are using the strong acid and the strong base, the nature of indicator which is used, it is different from when you are using the weak acid and the weak base. So for example, I can just give you an example so as you can understand. When you are using all of the components, they are strong. So let's say it is strong. Let's say it is strong acid, versus strong base. In this kind of titration, we have a wide range of indicator. Actually, you can use any indicator. Strong acid versus strong base, you can use any indicator. But when you are coming in the case, we have maybe strong acid then versus weak base. the case changes. Also, when you come in the case, we have weak acid versus strong base, then also the indicator changes. But if we have weak acid versus weak base, here we are saying no appropriate indicator, no appropriate indicator. Kwa hiyo this concept, they are very important for you to understand, very important, kwa sababu zitakusaidia sana wewe kuweza kuelewa different aspects. Kwa tuangalia hapo kwa haraka kuhusu hizo choice of indicators. Tuangalie kuhusu choice of indicators. So first, we have about 3 types of indicators in your level, and we use these even in the advanced chemistry. So we learned earlier that the estimation of the concentration of a solution of acid or base by reacting with the standard alkyl or acid solution respectively is known as titration. The end point of an acid-base titration is commonly determined using substances known as indicators, which usually portray certain characteristics, colors when in alkaline or acid solution. The indicators in acid-base titration must be chosen carefully because the choice of an inappropriate indicator will lead to incorrect results. The choice of an indicator is based on the strength of an acid or base involved in the reaction. Ndio maana tumeandika kuna strong na weak. Now remember, when we are saying strong, let me just define this in order for to simplify for you. A strong acid is an acid which dissociate completely when dissolved in an aqueous solution. So for example, when we are talking of the mineral acids, that means HCl. When you are talking from sulfuric acid H2, then SO, it is SO, then 4 sulfuric acid. And when you are talking of nitric, that means nitric acid, that means HNO, then 4. So all of these are 3, and even sometimes phosphoric acid, that means P, phosphoric acid P3, I mean it's H, it is H3, then H3PO, I mean PO, then it is H3PO, then 4. So when we are talking of these acids, they are all mineral acids, and because they are mineral acids, they dissociate completely when dissolved in aqueous solution. So these are just examples of the strong acids. But when you are talking of the weak acids, weak acids are acids which dissociate, which does not or which do not dissociate completely when dissolved in an aqueous, it is either solution. an easier way to define this, you can say when dissolved in water. When dissolved in water, if you don't remember an aqua solution, you can say when dissolved in water. So yeah, like that way. So examples of the weak acidity, we can talk of the weak carbonic acid. Weak carbonic acid is written as H, then H2, then CO3, weak carbonic acid. And all organic acids, they are weak acids. So for example, we have the CH3, acid is CH3, which is ethanoic acid, COOH. So this is ethanoic acid. It's also a weak acid. When dissolved in the aqua solution, they form an equilibrium solution. Kwa hiyo all of this unaweza kuona kwamba ni difference kubwa iliyoko kati ya weak acids and strong acids. Strong acids, they dissociate completely. Weak acids, they do not dissociate completely. Kwa hiyo that's the major difference between the weak and the strong acids. Now there are 3 common indicators which are used in the titration experiment involving acids and bases, namely methyl orange, litmus and phenolphthalein. So kuna indicator nyingine methyl red, yani hapo kemikali zinazo change color ziko nyingi, lakini the most common is tatu: methyl orange, litmus pamoja na POP. And this litmus is sometimes even in paper, litmus paper. Now you need to know, one among the very important thing to know for you in these indicators is their color, the color of indicator in acid, in alkaline and in the neutral solution. How does the indicator change color during titration? So for example methyl orange, its color is like yellow, or it's like orange. Actually its color is like yellow or orange. And in alkaline solution, the color is yellow. And in acidic solution, the color is pink. So when you are titrating, you are adding, let's say umeweka methyl orange kwenye alkaline solution. Most often hiyo ndio ambao huwa inakaa kwenye beaker, ndiko kwamba kunaweka indicator. So rangi yake itakuwa nini? Ni yellow. Kwa hiyo tutakiwa titrate mpaka rangi change, color change itokee iende kwenye rangi nyingine. Now remember, this table haija include color neutral. Nitakuja kuadi baadaye, nitakuja kuongeza. Lakini ninachomaanisha ni kwamba color ikisha change kwenda kwenye pink, that means umeshavuka the end point. End point color inabidi isiwe ile acidic, kwa sababu hatuhitaji kutitrate until the amount of acid increases, but you need to titrate up to the end point of our titration. Kwa hiyo that is a very important concept to know. Litmus kuna kwa red, ku blue, phenolphthalein, POP huko inakuwa ni colorless, huku pink. In terms of the indicators which you can use with respect to different types of solutions ambazo zinakuepo, in terms of indicators ambazo tunaweza kutumia, first tunaweza kuwa tuna strong acid na strong base. Kwa mfano ni HCl na sodium hydroxide and indicator. Tunaweza kuwa na weak acid na strong base, tunatumia kwa mfano acetic acid na tunatumia POP, phenolphthalein. Strong acid weak base tunatumia methyl orange, na weak acid pamoja na weak base tunasema no satisfactory indicator. So tunachokuwa tunakifanya in terms of procedure, tutaenda kuidiscuss kwenye session inayofuata. I don't want to make the session so longer and tiresome, but we discuss what is the basic procedures of volumetric analysis titration, and we will go even in some aspects of calculation, how we calculate light. But I hope you have understood about the concept of indicators na matumizi yake. Indicators, this table is very important. Sometimes teachers wanakuuliza swali kupitia hili table, wanakuletea kwamba utaje appropriate indicator ambayo utaitumia kwenye volumetric analysis tukiwa na titrate solution hii hapa na solution hii. So it is very important for you to know this table kichwani mwako. You need to know it, so you just cram and you stay with it. So thank you everybody. Don't forget to share this video and to subscribe to the channel. I will be launching our different sessions on the chemistry, biology, physics and mathematics for O level and A level. Subscribe to the channel, share the link of the video to your fellow students. Thank you everybody, and let me wish you nice studies.